Hey guys, so in this video, we're going to talk about gene expression. And in the next video, we'll talk more about the regulation of gene expression. So in this video, I want to focus on those other big points, right? The structure and function, those critical steps, those places where the process can get stopped very easily, and then also how this can relate to the fitness of an organism. I want to start with our diagram of DNA, right? And RNA will look very similar, it's just we'll have a slightly different sugar and different base pairs, or just one different base, the uracil instead of thymine. And so when we're looking at this, right, the main thing to know, right, you can, look up that it has these three parts to it: the phosphate group, the deoxyribose sugar, and the base. But the main thing to know is how the structure of this relates to its function, right? And when we're talking about that, I'm talking about things like the orientation, right? You have this 5' end and this 3' end of the sugar, right? That can so this is the 5' and the 3' end that bond, right? And so there's an orientation of DNA. And the reason that is really important is you can have two strands, right, connected to each other that are going in the opposite direction, and that helps the DNA get paired up, right? That's how you're able to pair these based on the structure of how they're oriented, and then also the purine pyrimidine structure of the DNA, right? You have the two rings matching with the one ring, right? That the way they bond together, that allows for the genetic code, that allows for those two strands, which end up making information a lot easier to keep because you have two strands, right? So the big ideas from the diagram here is how the structure of DNA, the way that it has this orientation, and also the way that the bases have these purine and pyrimidine structures that match up, that there are pairs. Those are two big ideas of how the DNA structure molecularly allows it to behave the way it does. Okay, so now we're at the central dogma. And the central dogma is how information travels in a cell. You have DNA, the double strand, right, and it has these base pairs. And then you have the DNA gets transcribed, right, and this is going to happen in the nucleus. Let's go to this. This was going to happen in the nucleus, right? You probably won't need to know that, but it may be important sort of conceptually of understanding how this works, right? And the translation will happen in the ribosomes. Okay, so the DNA, right, gets transcribed from 3' to 5'. So it gets transcribed 3' to 5', and as you can see, right, this mRNA strand is the matching strand, right? It's the corresponding, the complementary strand to the to this bottom strand, the 3' to 5' DNA strand, right? So where there's a T, it gets matched up with a U. An T got matched up with an A got matched up with a U. C got messed up with a G, matched up with a G. T with an A, etc. And then these come in pairs of 3, and those 3, right, they get transferred by that tRNA to form the polypeptide chain, which are, right, each individual amino acid, and then the ribosome is going to make these all go together and bond them together, right? And so that's how proteins are made from DNA, right? You have DNA, then transcription takes it to mRNA, then translation to a protein. And now we can look at the codon chart, right? And the codon chart, actually I'm going to pull this up a little bit. Here we go. So the codon chart tells us what codons, right? The combination of 3 nucleotides, which codons in mRNA make each amino acid. And there's a very interesting feature of this that I want to point out, is there's a lot of repetition, right? A lot of repetition here that, for example, if you have a C and a G, it doesn't matter what the next nucleotide is, it's always going to produce this ARG amino acid, right? Or if you have, right, similarly, this happens a lot. So you have a C and a U, doesn't matter what the next one is. Or you have a C and a C, it doesn't matter what the next one is. And it also works sort of if you look, if you have a U and an A or a U and a G, doesn't matter which one comes before it, it's still going to make LEU, right? And so the way that this is structured makes it so that mutations, right, a mutation where one nucleotide gets switched out for another one can actually be less effective, right, which helps the fitness of the organism, so it doesn't get, mutations that could end up killing it, right, because if you switched out, if you had a, let's see, you had T, or what would it be for LEU? You would have AAC in your DNA, right? But in a mutation, that got changed down. Instead of AAC, you got AAT. Well, you still have the same amino acid, so that mutation does nothing. And that's actually called a silent mutation. It just doesn't do anything, and that will help the organism, right, sort of manage the effects of mutations, the fact that there's this repetitive nature of the codon chart. And so then other types of mutations, though, can do some very serious things. So you have mutations where they will switch out one codon for another codon, right? Or one amino acid for another amino acid, right? So if you changed, let's look at where this ILE is on this chart. It's AUC. Well, what if it got changed to AUG, right? Instead of AC, well, now we're at methionine, right? So, and that's also the start codon, but it can also be a different amino. It's also an amino acid, and so that's that's sort of a problem, right? That you can have those mutations, one nucleotide change the amino acid, and we can see that in this problem from the College Board in the 2017 AP Biofree response. Also, just to note, there are some mutations that can also be really bad as well, not just changing a nucleotide, but eliminating one or adding one. That's called a frame shift mutation. Imagine we just got rid of one of these, got rid of that A. Well, now, all the codons after it are messed up, because now you're at UCU, and then GCU, and then AA, well, and then whatever comes after that, and it no longer stops. You're not at the stop codon. It changes everything after it if you subtract one or add one, right? It completely changes sort of the frame, the groups of three. It splits up those groups of three. But remember from our video on proteins, even one amino acid difference, that messes up the primary structure, and which messes up the secondary, then the tertiary, and then the quaternary structure. So one change in amino acid can, and one change in one nucleotide, right, can change the entire protein, make it a very damaging mutation, or very damaging effect. So let's look at this problem, and this problem will deal a little bit with this. I'm not going to pronounce that word, right? Gibberellin is the primary plant hormone that promotes stem elongation. GA3 beta hydroxylase is the enzyme that catalyzes the reaction that converts a precursor of gibberellin to the active form of gibberellin. A mutation of the GA3H gene results in a short plant phenotype. When a pure breeding tall plant is crossed with a pure breeding short plant, all offspring in the F1 generation, that first generation, are tall, right? And hopefully you've reviewed genetics. Again, I may get a video on that, but I can't promise. When the F1 plants are crossed with each other, 75% of the plants in the F2 generation are tall, and 25% of the plants are short, right? And so we have this universal genetic code, the codon chart. Okay, so the wild type allele encodes a GA3H enzyme with alanine, a nonpolar amino acid is the 229th amino acid. The mutant allele encodes the GA3H enzyme with a threonine, a polar amino acid, at position 229. Describe the effect of the mutation on the enzyme and provide reasoning to support how this mutation results in a short plant phenotype in homozygous recessive plants. And now, let's look at this. To start, this is a protein that promotes stem elongation. And if we switch from a nonpolar amino acid to a polar amino acid, right, that will affect the entire protein. That may make the protein, so the protein, just with that one change, won't work. Right? And so now we've gotten rid of a protein that promotes stem elongation, right? So it doesn't work anymore just because of that one amino acid. And so now we have a short plant, right? Hopefully that would make sense. Using the codon chart provided, predict the change in the codon sequence, right? Okay, so let's look at this. We had alanine in the beginning, so let's look at alanine. There we go, we're right here. And it got replaced with THR, right? So, well, let's look at this. All of these are THR, all of these are ALA. So what one was different, right? It looks like it got switched out in A for a G, right? So if we were to predict this, it would be A, so the mRNA went from A, C, anything, to G, C, anything, right? And so that was our change in the mRNA, right? Or you could look at what is the change in the DNA, and that you could write, and the answers that they were accepting that I saw in here, for if you looked at the 3' end, right? The 3' end would have been, right, if you just match it up, T, G, anything, 2. So this is mRNA. This is DNA. Right? So this is the change in the mRNA. The change in the DNA that could have been was from TG blank to CG blank. Right? One change in the DNA, one nucleotide difference, change the entire protein. You would have the 5' end here. They would have also accepted on their scoring guidelines, if you started with 5', it would have been anything, and then it's the complementary strand. So on 5', it would have been 5', then anything, G matches with C, and then A. And then anything, wait, sorry, yeah, 5', anything G, A, to anything C, G. Right? So again, just a single switch in either the 5' strand or the 3' strand, right? There's a lot of places it could have gone wrong in that DNA replication process. And so that's why also we have enzymes specially for trying to eliminate these mutations, because you can see one mutation, very damaging. And then describe how individuals with one heterozygous or two homozygous copies of the wild type GA3H allele can have the same phenotype. Right? And the answer to that would be, if you have two, right, you definitely have that gene that makes you grow, right? If you're heterozygous, right, if you're heterozygous, you have one copy of the gene that does work and one copy of the gene that doesn't work in terms of the protein, right? One protein works, one protein doesn't work. But as long as you have one copy that gets a protein that does work, you're all set, right? So that's what they were looking for there. Right? You could also argue it's a Mendelian inheritance pattern. So the heterozygous, the dominant, is the real growing one, right? They talked about that here. So I would probably give it an argument with the Punnett square, not a Punnett square, just ideas of Mendelian genetics. Okay, the last thing is sort of where gene expression is and how that works in the cell. Bacteria, right, and this is going to really have to do with fitness as well. Bacteria have everything. They don't have those specialized organelles. I mean, they'll have ribosomes sort of everywhere. They don't have a nucleus. And so they just have the DNA in a one ring along in the cytoplasm, and it's going to get translated, as we'll see in the next video, and it's going to make this polypeptide with the ribosome, which is a protein. And then the ribosome here, right, you have that, but now in a human cell or any eukaryotic cell, you have, you know, the DNA, it's in chromosomes, and you have these introns and exons, right? And so the only thing we really need to know about that for the AP is you have these parts, the introns that get cut out from the DNA, and the exons are what are left, right? You can see that there was processing that took out some parts of the DNA. And that is actually a way scientists, I don't think, fully understand what it does yet, and that's a lot of research going on. We understand a bit of it, but not everything that's going on and exactly why we have these introns and exons. But a lot of it could be to help our organism use the same strand of DNA to encode for a lot of different genes, right? Depending on how much you know, what parts you take out and what parts you don't take out, right? And so that will help us, as organisms, do more with less DNA, right? And we need to have a lot of genes to have a full eukaryotic organism, you know, survive. I mean, it takes a lot of proteins for that to happen. Bacteria require a lot less. Okay, so that is gene expression.